So my name is Thibaut Brunet. I was a PhD student in the EMBL from 2011 to 2016 in the lab of Detlef Arendt. And then I moved to do a postdoc at the University of California in Berkeley in the lab of Nicole King, and I've had my own lab at the Institut Pasteur in Paris since 2022.
How did you find the EMBL when you joined? I mean, how different it was from your university or from the type of institute you were used to?
Yeah, so I was not used when I first joined the EMBL. I didn't have a big experience of research. I actually first joined as a master's student in 2010. Or even 2009. So at the time I was 22-year-old, so I had very little experience of research, and I was so in a way it's maybe being spoiled, you know, having one of your first experiences being the EMBL. But I remember being really awestruck by the enthusiasm for science, how a community really there in the middle of the woods in Heidelberg, in this somewhat unlikely place, had crystallized, that really was united by a passion for figuring out how life was working. And beyond the science, of course, there was all this impressive logistics that is a little bit behind the scene, that is easy to forget about, because when it works well, it's invisible in a way. But still this impressive logistics, that means that a lot of things are possible very smoothly, like excellent technical facilities, access to the best experts in many techniques, simple things like a very smooth administration that doesn't stand in the way of science. And all of that, I think, even though it was made invisible, I could still feel that it was different from the other places, and that it really, everything was in the service of doing the best research we could, and people also had this drive in themselves. So I was very impressed, and I stayed there for 6 months during this internship, and I knew that it was a very good place to potentially come back later to do a PhD, and that's what I did. Yeah.
So how did you get to know the EMBL? How come that you decided to do an internship in another country? This is quite unusual.
Yeah, it was a bit of a fortunate accident in a way, or like I studied at the École Normale Supérieure in Paris, and one of the very good things about the ENS is that it was really preparing us to do research by encouraging us to actually go outside of the ENS and do research internships. And in particular, I was assigned a mentor, so a professor was helping me and giving me advice, and his name was Renaud de Rosa, and he was actually an evo-devo researcher, and so an expert in evolutionary developmental biology, which is also my field. And I had asked him for a name of good labs in Europe that he would advise to go to for internships. And one of the names he proposed was Detlef Arendt in EMBL. And I knew of some of Detlef's work at the time, but at the time I was reading scientific papers, but I was not necessarily connecting them to people or to places. So I realized when doing research about Detlef that, I know that guy's research, I know some of his past work, it's really interesting. And I also wanted to explore to go a bit outside of France. So I applied to Detlef's lab, and I wrote a very naive, very simple email saying, hey, I would like to do an internship, I'm interested in evo-devo, do you have space in your lab? And actually that speaks to Detlef's kindness and open-mindedness, that he answered my email positively, even though it was not one of these very nicely formatted, beautifully phrased emails that students, especially these days, send. Most students who write me are way better at asking for these things than I was at the time. But anyway, Detlef was, I think he could perceive some form of enthusiasm in spite of the very amateurish way in which I had written him. And then I was supervised by a PhD student, Antje Fischer at the time, who was also great in his lab, and that's how I discovered the EMBL.
At least they are not written by ChatGPT. I mean, I'm sure you were, at least expressing your genuine feelings.
It was very human. It was flawed in its humanity.
Okay.
Exactly.
That's great. So what did you do once in the PhD program? I mean, what was your science? Which kind of project did you engage in?
Yeah, so my interest was the evolution of muscle cells. I think I was curious about the evolution of movement, and that's a topic I've kept, thinking about ever since. And I actually was lucky to immediately team up with another PhD student, Antonella Lauri, who was a few years ahead of me. And she actually had identified a population of muscle cells in a marine worm named Platineris, which is the model organism that most of Detlef's lab studied at the time and still studies studies. And that population of muscle, she thought, had common points with an important organ in our body, which is the notochord, which forms in the embryo of all vertebrates and is in fact the scaffold around which the vertebrae then assemble. So if you want, it's a bit of a precursor to the spinal, to the spine. And she thought that basically this muscle that looked a bit like a notochord in terms of molecules and morphology was an indication that maybe our notochord had evolved by modification of an ancestral muscle. Now the notochord is not a muscle, it's actually cartilage. But she had this evolutionary hypothesis that maybe it had evolved from muscle. And since it was a bit peripheral to her main interest, which was really nervous system, but it was very close to my interests. And I think she was very generous in offering me to join this project and to collaborate and to do this together. And I've been very lucky actually over the years to have very good collaborators, people who were engaged into a shared quest for knowledge and not thinking too much about ego or credit, but just being really excited to work together. And Antonella Lauri was one of the first collaborators I had. So we did this project together on the origin of the notochord, and that was a long journey that took two or 3 years, but that was my first PhD project. And then after that, I also did a lot of projects, many of which actually failed. So science is also made of failure. And of course, if you look at the track record and the publications, you only see things that worked. But about half of my projects there just didn't work at all. So I tried following up on this notochord evolution project by doing some CRISPR knockout of platynerase genes. And most of the knockouts, for diverse reasons, didn't work at all. I was also a bit interested in mechanics of gastrulation in sea anemones, in Nematostella, which we had in the lab also at the time, and for diverse, and that's a very challenging thing to study. And I was trying it as a side project, and that it was not a good side project. It was too difficult. But I also had a project on the evolution of muscle cells more broadly, trying to understand how muscle cells had diversified when smooth and striated muscles split, and that actually was a paper. So that was my research. Basically, that was my research direction. But one of the things I really learned is to recognize when something doesn't work, and to accept at some point that even though you've invested time and effort in something, sometimes you have to give up and focus on what works. So I also learned that.
Yeah, I mean, this is an interesting concept. Because we know that if anything in science, most of the things are actually not working.
Absolutely,
rather than working. So I mean, maybe you pointed out the ability to actually see quite early what it doesn't work. But let me ask you this question. I mean, do you think there's some value in also describing what it doesn't work, if it's useful not to go in the same direction, or to just inform of what has been explored?
Yeah. So it's the age-old question of publishing negative results, right? And of course, when something, when you talk to scientists, very often people tell you, oh yeah, this or that technique doesn't really work because I've tried it in my lab, a lot of people have tried it in their labs, and by word of mouth, it doesn't work. But then if you're not plugged into the right circles, you might not know, and you might try it yourself and waste your time, right? It could be that 50 people waste their time independently on the same thing that simply is not working. I don't have a great solution to that. Of course, now, publication is no longer constrained by journals as much as before. We can just put stuff online. So there are a lot of platforms that are being tried for micro publications, and those are good, especially for sharing, I think, negative results. I haven't tried that very much yet, but I think that's for the future. It has huge potential. What we often do is that we put in the methods as much as we can of how we develop the methods and what we tried that failed. So if we tried a lot of treatments or approaches that turned out to not be the right way, we try to mention it very explicitly in the methods. We try to have a bit of a narrative aspect. So that's the way we found it to share negative results, but there are others. Yeah.
So and I mean, you mentioned collaborations and the nature of the work being always collaborative. But I was wondering, being close to your PhD classmate, was that an advantage? It was something that helped you in the PhD, and maybe you carried over later in the career? I mean, in other words, having an access to a new network of similar colleague it has been helpful? Yeah.
So EMBL is it's not just a place; it's really a community, and it's just striking how many people started their PhD in the same cohort as me. Are people I still meet in meetings everywhere, or I run around, and It's very, there's something special about EMBL in the sense that the communities are very cohesive. The PhD cohorts tend to stick very closely together. We go through this pre-doc course for 2 months when we start that is really, not doing research yet, but taking classes together, being together all the time, and there's also a very strong social aspect to it. There's something really about meeting your people, finding like-minded people who you can share the struggles with, the joys and the pains, and who really share your spirit. Many of the friends I made in EMBL are friends that who are friends for life. I know that, and even beyond close friends, there's really it's a diaspora, EMBL. Like people go to Heidelberg, but then they don't stay there very long. They either do a PhD or a postdoc. Even if they have a lab, usually they move, and then they spread over the world. But you do feel a connection through having that shared experience that also often means a shared spirit. And that actually everywhere I've been in the world, then when I went to Berkeley for my postdoc or back to Paris for my lab, and whenever I visit, there are EMBL people, and there's a sense in which we share something.
Yeah, no, definitely. I can relate because I was a student as well, and I can definitely share this feeling. Then you mentioned that, of course, you left the EMBL at the end of your PhD and you started your postdoc. So how did your science change? What did you get involved in when you joined Nicole King's lab at Berkeley?
Yeah, so I completely switched research organism and also experimental approaches. So basically, at the end of my PhD, I became very interested in the very beginning of animal evolution, and in particular, I had this interest in the evolution of movement, which means evolution of muscles, but also that's connected to the evolution of the nervous system that controls the muscles. And I was really struck by the fact that many of the molecules that are fundamental to how neuromuscular systems work were much older evolutionarily than animals. That if you trace them back in evolution, you find them in unicellular eukaryotes that are very distant from animals, and that's true of the machinery that implements movement, actin and myosin, that molecular motors that really allows muscle to contract. That's true also of many neurotransmitters that tell muscles when to contract or tell neurons when to fire. Often these actually are modified amino acids like glutamate or serotonin that you can find back in unicellular eukaryotes as well, and unicellular eukaryotes can also detect those molecules, presumably for another reason. They're not neurotransmitters, but they maybe signal that there is food around. And so I was fascinated by this idea that multicellularity had evolved in animal ancestors that went from being unicellular to multicellular, but it had been a lot of recycling and tinkering with things that existed before and that had another function in unicellular organisms. And so I became very curious about the closest unicellular relatives of animals, and in particular the immediate neighboring branch, which are the choanoflagellates. And I knew that there was one lab in the world, the lab of Nicole King, that was really developing them into tractable molecular research organisms. And so I wrote to Nicole King telling her, okay, I have this interest in the evolution of cell contractility, and I would like to pursue that. And she said, okay, well, I'm not sure choanoflagellates have much contractility, but you can come and research that anyway, and if it doesn't work, you could try something else. I mean, of course, I had to interview with her lab as well before being recruited. And that meant intellectually there was a continuity. It evolved from my PhD research, but in terms of experimental approaches, it was very different because choanoflagellates are microbes. So these are no longer animal embryos that I was used to, and you really work with the techniques and at the scale and at the speed of microbiology. So instead of having one animal in one flask, you have one flask in which you have maybe 1 million cells. Which comes with challenges for imaging. We do a lot of imaging, so they're much smaller. But also you have the power of numbers that comes with those large population sizes. And so it was very nice because I could still follow a little bit my passions on the topics I was interested in. But it was a complete switch in terms of approaches and techniques, and that's something important to do to embrace something completely new and to learn new techniques. Yeah.
I mean, what I find amazing of this is that you can see how, organization of life not only repurposes things, but also reproduces patterns on a scale. I mean, like, I guess in a single cell you learn that that machinery is used to react to the environment, but then when everything gets more complex and you have many cells and you have cell specialization, part of that specialization is geared towards the same. You're still reacting to the environment by building a nervous system and a muscle system, right? So it's pretty fascinating what you can get from fundamental research. So are you working now at different scales in your current research, or how did you move towards your independent lab?
Yeah, so we basically my directions in the research directions in my lab stem largely from the findings from my postdoc. And in particular, during my postdoc, we found by accident or serendipitously a new species of coinoflagellates that forms facultative multicellular colonies. So it can exist as a single cell, but it can also become multicellular. Okay. That was known about other coinoflagellates. It's not exclusive to that species. But what that species does is that those colonies also show spectacular collective behavior. So they form monolayers of cells, and those monolayers are curved in a cup, if you want, and those cups are very contractile. So they can change shape very quickly. Actually, what they do is that they flip inside out, they invert exactly like an umbrella. And so that's a behavior that's mediated by collective cell contractions. And what we are very interested in, we have several research directions in the lab, but we are very interested in really pinpointing that transition from one cell to many globally and trying to understand it in a holistic way, in a way, trying to understand what are the mechanisms that allow these colonies to form, what are the environmental signals that induce that transition, what is the adaptive advantage of switching in the natural environment of that species, and it's actually a species we can study in its natural environment because we can find it reliably in a similar place. And of course, we also want to understand mechanistically well how they come together, how they stick together, but also how is this collective behavior implemented? What are the cells doing? Are the cells speaking to each other? Are they exchanging simple or not so simple actually forms of signals? Are they repurposing modules that individual cells use to perceive their external environment into modules that are used for cells to now listen to their neighbors? So this is a very broad set of questions, but that's essentially what we are trying to address.
Well, that's exciting, and I'm really looking forward to see what your lab will find in the future. I mean, thinking back about the EMBL and Berkeley, very different environment, what are you bringing now to your lab from those experiences?
Yeah, so I guess I mean there are commonalities between both places. Both places really had a fearlessness and a boldness to ask big questions that I thought was exciting, and also at the same time extreme experimental and intellectual rigor. So that science really requires both legs of that dialectic, like being, brave and unafraid of dreaming maybe bold hypotheses, but also being completely unforgiving when a hypothesis doesn't work, testing it very rigorously and accepting the judgment of the empirical data and using the best techniques possible as well as possible. From EMBL, I think I keep really the sense of community. It's really a tightly bound community with a lot of shared resources, a lot of collaborations within the lab, and so on. And Berkeley was maybe a little bit more individualistic, like labs were working more on their own. But within the lab, there was really, especially Nicole King's lab, there was really this great spirit of helping each other, sharing a passion for the questions we were interested in, the evolution of multicellularity, and at the same time, while being supportive of each other's passion and drive, holding each other accountable in terms of, again, rigor and holding each other to high standards. And that's something that's very important in a small community like the coinoflagellate community. It's relatively few of us doing research on coinoflagellates. So if we want to maintain high standards of scientific rigor, in a way, there's no one else to hold us accountable except from our immediate colleagues. And so I think that's a spirit I think that we managed that in Nicole King's lab was very prevalent, that I think we managed to scale up at the scale of the community. So again, a transition, in level of organization, and that also I try to foster in my own lab.
Yeah, so no, that's great. And I'll finish asking you a question that occasion of the 50-year anniversary of the EMBL is what the EMBL means to you.
Yeah, so for me, EMBL is not just a place, but really a community and a spirit. And it's many things at once. It's people that I've been feeling close to and united in a common spirit with ever since I started my PhD here. But it's also a place that's never complacent and set in its ways. It's always a place that's reinventing itself and trying to anticipate or even contribute to the next big revolution in biology, and that included things like AlphaFold that EBI contributed enormously to in terms of predicting many protein structures. This includes things like the new planetary biology theme. So in a way, that's there is a constancy of spirit, of passion, of rigor, but also there is this ability to constantly reinvent itself that I think is one of the things that I love about EMBL.